All right. Okay. Hi, everyone. My name is Kevin Villegas Rosales. I'm one of the REPU co-directors, and I want to welcome you to the first part of the REPU Seminar 2023. And it's really my pleasure to be presenting in front of you. It was about 10 years ago when I did my REPU internship. And now 10 years have passed, and now I'm presenting not as a mentee, which we did at that time at Yale, but now as one of the REPU co-directors. So I'm extremely honored to be presenting in front of you right now. My background, I started my journey at REPU in the nanotechnology branch of REPU back in 2013. And over the years, I have been involved in nanotechnology and in physics. So that's where I come from. And the reason why I tell you this is because in the directorship of REPU, we have 3 different people. Besides me, there are other 2. One is Jorge Rua, and the other one is Brenda de Acuña. And each of them actually have been part of REPU, and they each belong to different branches of REPU. Jorge did his internship with biology, and Brenda with chemistry. Okay. So first, I will do a few minutes introduction before we start with the presentations of our mentees of 2023. And the virtual part of the seminar will be running for about 2 hours. We'll have 3 mentees presenting today. And before closure, we will have Jorge Rua, one of our co-directors, presenting some of his recent work. And then we will conclude. And I would also like to tell you that the second part of the seminar will be happening this next Saturday. And it will be hybrid. There will be virtual presentations as well as in-person presentations. And we will be very happy to be seeing you there. Okay, so what is REPU? I first want to say, what does REPU means to me? This answer has changed over the years. And as of now, to me, REPU is like an act of kindness and love. There are dozens of volunteers every year preparing the stage for what REPU is during the months of January and March each year. And I'm referring to dozens of volunteers in different parts of the stages of the preparation, from admissions to paperwork and during the internship. And they do change. REPU, it is a program that focuses on improving the capabilities and knowledge of undergraduate students from Peru. Each of the undergraduates have a unique opportunity to do a stage or an internship in a well-renowned university, in a research laboratory that is very well equipped with not only physical equipment but also with the knowledge to do advanced research. And this happens across the different branches of REPU, like biology, nanotechnology, chemistry, computer science, ecology, and physics. Okay. Over the years, we have had Peruvians, undergraduates, participating in different universities around the world. Here, we're listing a few. The REPU has taken place in North America and in Europe. And when I say North America, I'm referring to United States and Canada. And in Europe, it really has been all over the place. We have had students in Spain, France, Germany, Denmark, and this year we have people in Sweden. So it's really a very diverse program. So here we're listing only a few universities. It's not supposed to be a complete representation of all the universities that have been able to help Peruvian undergraduates. And one thing that I want to share with you is that this year, the 18 participants of the REPU program, all of them went to Europe. Unlike previous years where the program, we have had people participating in the United States and some of them in Europe, for example, this year everybody is focused in Europe. It is a unique time. And it happened that during the preparation towards the program, there was some obstacles that make it a little bit more of a delay to proceed with internships in the United States. So we chose to make it happen only in Europe. And we really, it has been amazing that, and like any other year where maybe we had 20 participants distributed between North America and Europe, now all of them went to Europe, which means that there are a lot of volunteers and willingness to help different Peruvians in Europe, which makes me think what's going to happen next when North America and Europe return as host places for undergraduates, and it makes me wonder if we're going to maybe achieve 40 Peruvian undergraduates next year. Since we can send 18 to Europe, maybe we can send 18 to North America, and then we'll have many more. Okay. So I want to say a few numbers about REPU. We have hundreds of applicants each year, which includes all the different scientific branches. And it really boils down to, like, the admission rate is on the order of less than 10%, which is, it's very small, it's very competitive. But really, I have to admit that I participate in the admissions of a few branches. I don't participate in all the different branches, but I sincerely can tell you that the students who were chosen have demonstrated a strong background and willingness to do scientific research, and that's why they were selected among their peers. And we put our complete trust in them, that they're going to excel in their January to March internship, but it really goes beyond that. The January to March internship is just the very tiny but extremely important step towards a strong scientific career, whether that is being part of a university, a national laboratory, or in industry. I would like to mention this again. The REPU program is currently has 3 co-directors. Jorge Rua, that is currently PhD candidate at Vanderbilt University. Second one is myself. I'm currently employed at a startup called Quantum Machines. And Brenda de Acuña is our last co-director, who she is now working in the UK Center for Ecology and Hydrology. We also have an advisory board, and the advisory board meets with the directors once a month at least to discuss relevant topics into what is the current status and the direction of REPU. Our current advisory board is made of Abel Alcázar, Kenji Saito, Paola Moreno, and Edson Bellido, and these people have been really key participants of the REPU program in the past 14 years. They have played a very crucial roles that have made a very strong impact in the growth and development of REPU. One thing I want to say, which is very special this year, is that thanks to Artificio, which is the startup founded by Arturo Deza and his colleagues, the computer science branch of REPU program received funding directly from them, which made it, I think, significantly much more feasible for the internships to occur. Because I think it's important to mention that even though the REPU program is made out of volunteers and offers great devotion to the undergrads that participate in the program, at this moment in time, we are making progress towards increasing the amount of funding we can provide to our undergraduates, but it's far from being thorough and comprehensive, which makes that our undergraduates also make great efforts at length to be able to secure their own funding for the internships that includes airfare, housing, and a stipend for food and transportation while they are abroad. I want to give a special thanks again to Artificio and Arturo Deza and his colleagues for supporting the and complementing the education of Peruvian undergraduates in the computer science branch of REPU. Okay. So in my last minutes, I want to make a few comments about what is next. But before I do that, I want to say that the REPU was founded in 2008 with the branch of biology. Okay. Yeah. And it started with one mentor and one mentee. And it ran for several years as REPU was only focused in biology. And in 2012, 2 new branches were added to the REPU program. Those were nanotechnology and chemistry. So by 2013, I guess I was telling you at the beginning, 10 years ago, when I was presenting as a mentee, that was the first time that nanotechnology and chemistry joined biology in the REPU program. Soon after, in 2014, the physics branch was open and has been working since then. And could you please mute yourself? I think I'm hearing some background. Thank you. Okay. So as I was telling you, in 2014, the physics branch started. And in 2016, the ecology branch initiated and has been running since as well. Our most recent addition to the branches of REPU has been in 2020, the computer science branch, which had a unique start. It started as a purely virtual internship. And it had to do with the context of that year, which many of us remember is not so long time ago, that was the year when COVID started. And since 2020, computer science has been going, and it's a very strong part of our program. Okay. Currently, we have several STEM branches, and I wouldn't be surprised if we keep adding more with the years ahead of us. And it's going to be very exciting to see that. Okay, so what's next before I wrap up? Okay, so the thing is that over the years when I talk to mentees, I realized that everybody does January to March work, and then they do not always hear from their environments what is going to happen next. Many of you are doing internships while you are in maybe your senior year or a recent graduate from the university. So maybe most of you have a very strong desire to pursue graduate studies. So that's maybe one thing that's coming next. Not all mentees of REPU started graduate study. There are a few that had taken a personal decision to stay in Peru, and they are actually active participants of REPU, but they chose to pursue careers in Peru. But the majority of our mentees pursue graduate studies. And maybe the question, what's next, refers to what is the gap between performing or doing a REPU internship and a graduate studies. So the moment you will return home after these internships, you will start preparing towards the application of grad studies, which is made of taking tests for showing your English proficiency. The test may include as well some, depending on to which continent you're applying to, it can be made of mathematical and language abilities. It will be important for you to gather letter of recommendations as well. And you will have to write personal statements to complete the application to your graduate studies. And I want to, before I close, I want to tell you that over the next period after this March, that you rely on the mentors that you have made during this internship, and that you have been granted and now own a place at REPU, so that at any time you can actually ask for help to anybody that is part of the community. And it's not going to be only for your graduate studies applications, but really at any point of your careers, whether that is to pursue graduate studies or even after finishing graduate studies, what is going to happen next, which could be pursuing a postdoctoral position or an industry position. And with that, I would like to close my part. And in a few minutes, we'll start with the presentations from the mentees. Thank you. Okay. Thank you, Bernardo. Now, we would like to start the presentation from the mentees. It starts with with Bernardo and Christian. They will both be co-presenting, and they belong to the ecology branch of REPU. Thank you, Bernardo. You can start at your own time.
Great. Can you hear me or?
Yes.
Okay, great. So good afternoon, everyone. Now we're going to present our research project, which is titled Pollen Transfer Patterns in Bus-Pollinated Solanum, Influence of Floral Morphology and Pollinator Size, which was carried out at the Vallejo Marine Lab at Uppsala University by Ari Vasquez and Bernardo Garcia as part of the REPU program.
Hi guys, first we need to talk about what is bus pollination. And this type of pollination is a very curious pollination system where the pollinator needs to produce vibrations, or also called sonications, in order to release the pollen from the anthers. Now, why is this process important? Approximately between 15,000 and 20,000 species of plants rely on this mechanism, and in this group are included some of the most important agricultural crops, such as tomatoes, potatoes, eggplants, blueberries, and etc. And here we have a diagram that shows all the process. At the beginning, the pollen is inside the anthers, and in the case of solanum flowers, the anthers have a pore in the tip of it, and this structure is called poricidal anthers. Then the pollinator visits the flower, and it curves its body around the anthers, like it's shown in the picture, and start to produce vibrations to release the pollen. So pollinators also needs to exhibit the buzzing behavior. For example, bumblebees and carpenter bees can produce these type of vibrations by using their flight muscles, while honeybees can't do it. After the pollen is released, a part of the total pollen doesn't land on the pollinator's body. This is not considered in the process of pollination, and it's the pollen wastage. Also, there is a part that it remains inside the anthers, and there is a part that is collected effectively by the bumblebee, and the pollinator then grooms its body, and this pollen is stored in the curbicula, or also called pollen baskets. But there is a portion of it that is not groomed and stays in the body. And this fraction is the one that potentially can be deposited on the stigmas. However, bus pollination is affected by floral and pollinator traits. For example, buzzing properties, if the pollinator produces vibrations with a higher intensity or with a higher duration, more pollen will be released from the anthers. Also, the architecture and the size of the flower have a crucial role, because both determine the handling that the pollinator has during the visit, and also the transmission of the waves on the anthers or in the flower. And finally, we have some behavioral patterns like grooming. For example, if the pollinator has an intense grooming in its visit, more pollen will be collected in the corbicula and not be available for pollination.
Okay, now that we know what is bus pollination, we can talk about the process of pollen dispersal. This is basically how the pollen from a donor flower is going to be distributed among the flowers that receive this pollen. For this, we can make a small example. We have a donor flower here. We're just going to focus on the pollen from this flower. So the pollinator visits a flower and transfers this pollen to the flowers that are subsequently visited. But of course, we don't expect that this pollen is going to be distributed evenly among these subsequently visited flowers that are called the recipient flowers. But most of the pollen is going to be deposited on the lower order recipients, the first recipients. And it's, of course, going to decrease as the order of recipient increases. This pattern can lead us to a pollen deposition curve, which is represented here. Here we can see the proportion of the pollen from the donor that it's going to be deposited on the different order of the recipients. We see that most of this pollen goes to the first order recipients, leaving just small amounts of pollen to higher order recipients. So we see an exponential decrease, but with a really long tail that sometimes can represent the long distance pollen dispersal process. However, measuring this process has some practical problems. This is why pollen dispersal curves are scarce in the literature, but they're really important because they can explain different patterns in the reproductive cycle of plants and the genetic structure of populations. This way, we established 3 research questions. First one is, what is the efficiency and distribution of transferred pollen from pollen release to pollen deposition on stigmas along subsequent visits? The second one, species with different anther morphology exhibit distinct pollen deposition patterns. And finally, is pollen deposition affected by pollinator morphology? In this case, we measured the bee body size.
For our research, we use 3 species of Solanum and 1 species of bumblebee, that is Bombus terrestris, and they were from artificial hives. And we use Bombus terrestris because of its high variation in size, so we can see if the size has an effect on the pollen deposition. About the Solanum plants, these were raised on greenhouse conditions in Uppsala University, and the 3 species of Solanum have contrasting morphology. For instance, Solanum sessilifolium has 3 anthers, so the anthers are independent from each other, and it's no heteranthers. On the other hand, Solanum dulcamara has joint anthers. That means that anthers are forming a structure called anther cone, and it's also no heteranthers. And finally, we have Solanum rostratum that has 3 anthers and heteranthery. That means that has 2 types of anthers. 4 feeding anthers that are pointed by the blue arrow, and these ones are focused on providing pollen to the bee as food resource, while we have also the pollinating anther, that is the gray one from below and is pointed by the red arrow. This one focused on deposit pollen in the pollinator's body to ensure the pollination process. And also, Solanum rostratum has an antiostole. That means that the style of the flower could take 2 positions inside it. It could be on the left or in the right of the pollinating anther.
Before getting into more detail about the experimental process, we have to think about how can we do experiments with bumblebees in Sweden in the middle of winter, right? So behind all the experimentation process, there are lots of extra work that we have to do in order to take care of the bumblebees, of the hives, and also of the flowers. First, we need a home. As Ari mentioned, the bumblebees are bought from commercial hives that are produced for crop management and are connected to a flight arena. A box. One of the sides of the box is transparent, so we can see everything inside. It also has artificial illumination, so we have light. And here's basically the place where the bumblebees live. They forage, they eat nectar, they eat pollen. They do everything between those places in the hive and in the flight arena. And we also perform the experiments here. Also, we need food. We need nectar and pollen. We made artificial nectar by mixing sucrose and water and use these special nectar feeders for bumblebees that we have in the lab. And also for the pollen, we mixed pollen and nectar to make these pollen balls that are given to the hive couple days. Finally, before performing the experiments, we have to get the bumblebees used to visit flowers. Before this, it's called the shaping process, this initial process of presenting the flowers to the bumblebees so they can forage, they can start buzzing, because before they haven't ever seen a flower. And this is really important. Otherwise, you won't be able to perform your experiments, and your bumblebees will never visit your flowers in the experimental trials.
So about our trials, we use 2 hives, and the hives were connected to the flight arena, as Bernardo said. And for each trial, we presented a sequence of flower to a single bumblebee. This sequence consisted in 1 donor flower and 4 recipients from the same species. In the recipient flowers, the anther pores were glued, so no pollen is released during the visit. And we also add this glue on the donor flowers, but only in the base of the anther, of course. And this is to avoid any effect that the glue can have on the trials. The trials consisted in setting the donor inside the flight arena with this black surface that it's on the photo, and then we release the bee inside the flight arena and wait until it visits the donor. After 1 single visit, and that's very important in our trials, we took out the donor and put out the first recipient in, and we repeat this for the other recipients too. And also we measure the duration of each visit. After the trial, we collected the pollen remaining on the anther, so we collect the entire anther from the donor flower, and we also collected the pollen from the black surface. And this is like a proxy for pollen wastage. From the recipients, we collected the stigmas to determine the pollen that was deposited in 1 visit. Finally, we collected the pollen from the body of the bee. That is the available for pollination, and pollen from the corbicula. We also take measurements of the bee, and we collected all these pollen using fuchsin jelly, and we store them in Eppendorf tubes.
After obtaining samples from our trial, we proceeded to the pollen counting process. For the samples of the stigmas that have a countable amount of pollen, we just mounted them directly on slides and visualized them under the microscope so we could count the number of pollen grains. In case of other samples, such as the anthers that can have up to 500,000 pollen grains, that's of course not an option. So we used an hemocytometer to just measure the amount of pollen in a known volume and extrapolate that to the whole sample. Here you can see one of the collected stigmas under the microscope. And these are the pollen grains that are deposited on it. And here's Ari during the pollen counting process. And pollen counting takes time because some samples of the stigmas can have up to 2,500 pollen grains. So it's a really laborious process.
And for the data analysis, we will compare the efficiency of Bombus terrestris as his role in pollination in the 3 species of Solanum using pollen deposition curves. And we will also build a GLM model to analyze which of the explanatory variables, those are number of recipient flower species, bee measurements, and the duration of the visit, has a major effect on our response variable that is the pollen deposited on a stigma. We are adding as random effects the BID and the hive ID.
Right now, we are in the pollen counting process. We still have some time to finish, and we have counted only the pollen that has been deposited on the stigmas, but we have a lot of samples that still remain there for counting. But what do we expect? First, we expect the typical decreasing that it's observed in pollen deposition curves. So as the number of recipient increases, the amount of pollen grains deposited in the stigma will decrease. Additionally, regarding the floral morphology, we think it's going to have a significant effect on the pollen deposition curve. From this species, the most interesting one is Solanum rostratum. As Ari mentioned earlier, this species is heterantherous. This means that it has two types of anthers, the feeding anthers and the pollinating anther. The pollinating anther makes this a strategical placement of the pollen on the pollinator's body. So the effect of the grooming is this process of collection of the pollen that the pollinator does, is going to be less intense. Usually, when the grooming effect is more intense, pollen deposition curves are steeper. This way, if the pollinating anther places the pollen strategically on the pollinator's body to reduce the impact of the grooming, we would expect a less steep curve and a more even distribution of the pollen on the recipients for Solanum rostratum. Additionally, the size of the pollinator can have important implications on its foraging capacity, also its buzzing capacity. So we expect that there will be different patterns in the related to the pollinator size. Also, it affects the fit between the flower and the pollinator.
We also have some preliminary results, but again, we haven't counted all the slides, so we only have like a partial view of our data, and these figures are more like visual representations, but we haven't used any statistical test yet to prove if there is any difference between them. The first figure shows us the pollen grains deposited on the stigma per recipient and per species, and this is in absolute value. So we can see that in the case of Solanum rostratum, less pollen is deposited on the stigma. That could mean that Bombus terrestris is not as good pollinator as in the other species, or for example, Dulcamara, that it's like a little has more pollen. Or also that could mean that just rostratum produces less pollen. In both Solanum dulcamara and Solanum rostratum, we can see the pattern we expected about the pollen deposition curve, with a higher amount of pollen in the first visits, while less amount of pollen in the last ones. However, with Solanum cisembrifolium, we can't see this pattern actually. So we thought that probably we have the hypothesis that when we were gluing the pores, we noticed that pollen was still being released, and we realized that in some flowers of this species, the longitudinal dehiscence was still present. That is very interesting because it is supposed that Solanus cisembrifolium only has the poricidal anthers, but in this case, we also found that has this longitudinal dehiscence. We still don't know if this is a mutation of this population we used, or maybe it was some kind of mechanism to ensure the pollination, but it was pretty cool. We found this. And in the other graph, these are the same variables, but with proportions, so we can compare the three species. And we can see more clearly this pattern in Dulcamara and Rostratum. And Rostratum kind of shows a more steeper curve. And also we have the same problem with cisembrifolium. We can see really a pattern here.
So what's next? We haven't yet finished our project. We still have to count pollen from the anthers, pollen from the pollinator's body, and the pollen wastage. This will give us a better view of the pollen dispersal process, and of course, additional information to interpret our results. After that, we're going to go for the data analysis. We're working on that partially. And with Mario, with our PI, we're looking forward to eventually publishing it. And regarding our ideas, Ari and I are really interested in pollination ecology. So we really want to keep working on this area because it's amazing and really interesting. So no, we don't have results yet. Still, we have 3 take-home messages. The first one is that we talked about bus pollination. That is an amazing process that occurs between bus pollinated plants and buzzing pollinators or pollinators that can emit sonications. These pollinators have to vibrate the flowers in order to extract the pollen. Additionally, we also talked about the pollen dispersal process that plays a fundamental role in the reproductive cycle of higher plants and can have an important impact on population genetical structures. And finally, that floral and pollinator traits play a major role determining the dynamics of these plant-pollinator interactions. And this is one of the points that we want to assess here, how these floral and pollinator traits affect the pollen dispersal process in bus-pollinated plants. So here you have a really short video. It's just a bumblebee visiting flowers that we recorded during our experiments. This is the donor flower, so it can release pollen, and this is the black surface that we use to collect the pollen wastage that you can see here, how the pollen falls from the flower. So I hope it was a really interesting talk for you.
So in this research, we received help from a lot of people, and wouldn't have been possible without the support of them. And for example, Mario, our PI, that received us in his lab and always have the time to guide our ideas. Also, the other members of the lab, Elodie Morel, that is the third member of this project, and Daniela Escacabarosi, Pilar Herrera, that is our mentor from the program and made this new experience easygoing for us. And finally, Uppsala University and the REPU program for giving us this amazing opportunity. So thanks to all of you. And thanks for listening.
If you have any questions, it will be amazing to answer them or talk a little bit about them.
Thank you, Bernardo and Ari. Yes, does anybody has any question? Jorge?
Hello, great talk. I was wondering if, besides the bumblebee, do you guys know if there are only pollinators that prefers the free anthers than the joint anthers? Is that something that has been seen with other pollinators?
Ari?
Oh, yeah. We don't know actually if there is any preference from choosing free anthers or joint anthers, because they want the pollen as a resource. So I think if they gather pollen, it's the only thing they care about. Also, there are some other bus-pollinated plants that also has nectar, so they can collect also like this nectar from them. I don't know. Have you heard of like a preference between both flowers, Bernardo?
Yeah, I think it not specifically in bumblebees. If we talk about in general, bus pollinators are really variable. It's going to depend a lot on the feed or the characteristics of the flower for each pollinator. Different types of pollination have different ways of manipulating the flowers, for example. So it's going to depend a lot on specific pollinator and the specific plant species. It's a really complex interaction. So it's not a specific preference for type of flower in general.
Yeah, I would assume that the insect morphology also might take into account if they can hold a specific type of anther.
Yeah, that's a really good point, because, for example, I'm gonna go back to Solanum rostratum. Is really interesting because we talked about the feeding anthers. So basically, the pollinator focuses more on these anthers, are more visible, more conspicuous, and try to take the pollen from those anthers because they feed on pollen, but that's not convenient for the plant. This is why some plants have developed a pollinating anther that places the pollen exactly in the back of the pollinator, and it fits really well with the pollinator morphology. But of course, for some pollinators, smaller pollinators won't fit really well, depending on the size of the flower. So it's going to be really variable, and it's also a really interesting process, this feed between plant and pollinator.
Okay, thank you.
Anybody else has any other question? I have a question myself, and I don't have any biology background, but I think these questions to me are very interesting. Like the first one is, how do you control for not being outdoors? Like, like the bee out in the wilderness may have another sunlight conditions, humidity conditions, wind conditions, predators, all sorts of things that are so different compared to being in the lab. And I think it's very interesting that you can do these studies in the lab, but how do you control for this, and how do your results translate?
Yeah, you mean how does this resemble what happens in an actual ecosystem, right?
Exactly.
I think with this general ecosystems and this type of interactions being really complex, we have to go to the basics and study like each part. For example, at least in bus pollination, there are different approaches. So since what happens in reality is so complex, you try to control for most of the things that you can, so you can understand the basic processes, and then relate, for example, the pollen deposition patterns with other floral characteristics that can be studied. But apart from that, because you cannot include everything in one study, there's no way you can control for everything. And going to the basics, and from that basic things start building up to understand a super complex system. So yeah, it doesn't resemble what actually happens. For example, we glued the recipient flowers so they cannot liberate pollen because we just wanted to count the pollen from the donor, and that is obviously going to affect the behavior maybe of the pollinator. But there is no other option of approximating to it. Other options include genetic methods to identify the where the pollen comes from, but with the genetic tools, and that's of course not viable. It depends a lot on, yeah, it's quite complicated, and it's never going to resemble exactly what happened. The starting point to understand complex processes.
Thank you. That's very insightful. I think we can close your presentation and move to the next one.
Okay,
thank you so
great.
much, Bernardo and Ari.
Okay, thanks.
Thank you for your questions and for listening.
Of course. Thank you. Camila, yes, you're ready.
I'm ready. So please tell me if you can see the screen.
It's loading. We see the presentation mode, but I also see the bar, like the icons on the bottom.
Oh yeah, it always shows.
Okay, no problem.
Okay,
Then
so
I can see the presentation and we can hear you well.
okay, so I can start now, right?
Correct. Yes.
Okay. So hello to everyone. Thank you for listening to this presentation. So I'm going to start. My project I developed during this internship is entitled Exploring the Phylogenetic Relationships within the Motacilla genus using Mitochondrial DNA. And I did this project during my visit to Uppsala University. So these beautiful pictures are some of the Motacilla species we're going to talk about. So first of all, a little background. We need to remember that sexual selection is a potent diversifying force in many animal systems. And in novel science can drive continuous and often rapid divergence in sexually selected traits between populations. In birds, most important sexually selected traits are plumage color and pattern, and also the singing. So here we can see a rapid divergence of Motacilla alba species into many different subspecies that have very different plumage colorations. So there has been detected that there are very few genes related to this divergence in plumage. And when exploring those genes, there has been detected a little genetic differentiation. So why may be that? There are two most accepted explanations for that. The first one says that there has been a very recent divergence in this species that have a very, very distinctive variation in plumage coloration. So as the divergence has been so fast in time, it has not been time long enough for this differences to fix on the genome. Other explanation is large-scale introgression, which means there has been reproduction outside the species level. What animal system can we use to study this? It's very helpful to study genera with widespread species complexes, like the Motacilla genus. Yeah, we have here many different species that have radiated very fast, and we can see that they have like many subspecies for each species, being Motacilla flava, the yellow wagtail, the one that has most subspecies, being 13 at all. So a first bit of Motacilla flava, we have to talk about them. They are a Palearctic bird species. They are very widespread, as you can see in the map. Some of them are sympatric. You can see the overlap in the yellow wagtail's breeding ranges, for example, in this zone and in this zone. And also something is very important is that they are migratory birds. And they are fascinating, but why to study them? So there has been detected mitonuclear discordance. Here we can see that Motacilla citriola and Motacilla flava are in a monophyletic clade, which means they all descend from a common ancestor. But when we explore this phylogeny and divergence, tree has been made with mitochondrial and nuclear information, and we can see that the Motacilla genus, all in all, has diverged very rapidly in time. But what happens when we explore a tree that is only made of mitochondrial DNA information? We can see that these species are separated, they're aggruppated in two different clades. For example, here we can see clade X and Y, and also separated clade C. This is a dispositioning polyphyletic, which means there are species separated in different clades, and you do not include all the descendants of a common ancestors, because they share a common ancestors, but we don't include all of the other individuals. So this is an example of a polyphyletic tree. So here we can see the many different variable phenotypes of Motacilla species. So there has been said that relationships among these species are really unclear and they are in urgent need of re-examination. So in my project, I'm going to be exploring phylogenetic relationships within the Motacilla genus using the whole mitochondrial DNA, the whole mitogenome. So first of all, I'm going to talk to you about a little bit about my methodology. First of all, I had to process the data my PI gave me. So I had to align the sequences to a reference genome. Later, I had to transform this SAM file to a BAM file, and then I had to do a process called variant calling, in which I'm only going to stay with those sequence reads that have polymorphisms. So this is a variant calling workflow that I follow, and I use three new programs for me. The first one is Bowtie, SAM tools, and BCF tools. Next, when I have all my sequences ready, I proceeded with the alignment using the program ClustalX. Later, to check the quality of my data, I had to explore the sequence's completeness. So this means that in some parts of my sequence, I will have undetermined characters, which will lead me to the missing percentage. And this is very important in order to choose which sequences I'm going to use for my analysis or not, because it's not very useful to use sequences that have most of their characters missing. So for this, I use the program AMAS. And last but not least, I had to remove those gaps that are not useful information, because it will be like comparing nothing to something. So I had to remove them. And later I generated multiple trees to evaluate which one was the better. So for removing the gaps, I use gblocks, and for making the trees, I use IQ3. And I did this processing for over 90 samples, which was a lot of work. This is my latest tree, and this is a maximum likelihood tree. Overall, the branches, the nodes have a really good support, because when you do these phylogenetic trees, the programs run them over and over again. In my case, I ran them for 1,000 repetitions. In most of these repetitions, the disposition and topology of the tree were most likely the same, which means that my nodes have really good support, and this is a really good supported tree. So I made this tree with 55 sequences, because when I was checking the completeness of the data, I decided to use a threshold to stay with the sequences that have less than 10% missing data. So yeah, so it's really important to have a tree to compare the phylogenetic relationships. So here we can see how my tree is different to this first one. Yeah, there is supposed to have two small clades, which have Motacilla flava, Motacilla citriola, which is correct. But here I found some difference of the disposition on the topology of the tree, because Motacilla alba species shouldn't be right here, but which is very weird since I have really good support on this tree. But my data is very different to the one that Harris and collaborators used. So this will explain the missing data. Also, maybe I have to have a more strict filter in order to have a tree more similar to Harris. So this has been my results. So I have some take-home messages for you. First, we need to remember that phylogenetic trees are hypotheses. So they can differ when using different data sets, which is really valid. Yeah, but it's also very useful to have a reference tree to compare the phylogenetic relationships among species. And also we have to remember that data quality affects the topology of the tree. For example, in some cases I have polytemies, which means that I don't have dichotomic relationships, and these things need to be like repaired. I need to improve them. So yes, but at last I want to thank everyone in the REPU program for allowing me to have this wonderful experience. At Uppsala University, I was part of the Department of Ecology and Genetics and of the Animal Ecology Office. I have to thank a lot Per Alstrom and Lois Franzilek for supervising and teaching. Most of these programs, I didn't know how to use them before, and it was really nice to learn how to use different programs to the ones that I knew. And Pilar Herrera and Jimena Alba for their help and support during this whole trip. Thank you so much. So I wonder if you have any questions for me. I'll be happy to answer them.
Thank you, Camila. Does anybody has any questions? Jorge?
Of course, I do have questions. It was great. Congrats, Camila. I like your presentation. Well, it made me reminded, you said that comparing the two trees might be different. Do you know if they use a different sequence than yours? Because you used whole mitochondrial sequence. Is that the same case of Harris? And a second question, do you think this kind of research can be done in Peru as well?
Yes, thank you for your questions. Yeah, as I remember, Harris and collaborators only use some of the genes. For example, in many studies using mitochondrial genes, the most used are ND2, COX-1 and cytochrome B oxidase. The second one is that if we can do this research in Peru, yes, definitely yes. I think there has been done here as well, and it will be nice to explore our diversity using these phylogenetic networks, but they also require to have really good funding to do the lab processing. For example, in this case, I did not participate in the lab because the DNA extraction and evaluation of the quality of the DNA was done before I got there, and they sent all this DNA to sequence, so I directly work with the result of that. But yeah, I think in Peru you can make this as well.
Hey, thank you.
Thank you, Jorge. Bernardo?
Hi, Camila.
I
I have a question. I think it's important. This biology.
I'm sorry, I can't hear you.
Okay. When
Now,
you perform this type of analysis, you have a lot of trees, but you have to choose one of those trees.
yeah.
What do you base your decision on? Just on other trees that have been done before and other parameters of the trees, or?
Yeah, that's a really good question, and I'm glad you asked. So we have something called bootstrap, which are repetitions of the tree. For example, in my case, I told the program to do the phylogenetic reconstruction for the minimum is 1,000 times. So they make the tree all over and over again using the program, and they show the tree that was that has the most repeated results. So you can do like many of these repetitions. For example, I use 1,000, but in other analysis I made in the past, I used like 75,000 repetitions. So you can make a summary tree out of this. Yeah, and if the program does not give it to you, you can make it using other programs. And it's a summary tree which with the results that repeat all over and over again, which are the ones that have most support. You can also change the parameters of the tree according to some things, you know, for example, the mutation rate of different genes. You can use that to change the parameters of the program in order to get a more specific tree. But yeah, you can do that. The program usually tells you their thresholds, so you can keep playing with the parameters until the program tells you that yeah, your parameters are good and you can follow your analysis. Yeah, but it's a really good question. Thank you.
Thanks for your answer.
Thank you. Camila, I have a question. It's a little bit general. So this bird, its home is Europe and Africa, right? Like Northern Europe.
Yeah, it has also been detected in North America.
Okay. So actually, that's a little bit of my question. Like,
when you make your question, thank you for your question, and I also think it's very nice to talk about their distribution, because as we can see, they are widely distributed among Europe and also, as I told you, some of North America. So it's very useful to see the speciation continuum among them, because they belong all to the same species, but they have diverged into subspecies, which have very different phenotypic patterns on coloration plumage, which is really nice.
I see. And and they are not found in South America?
No.
What is what prevents them to migrate southwards?
I don't think that I'm not sure if something prevents them from coming to South America, but I think their distribution is so wide during Europe, because in the last glacial maximum, they had to find refugees in different parts of Europe, and they stick into these refugees, and that helped to differentiate over the time. Yeah, that is why there's overall wide range, but I don't know why they don't come to South America, but yeah, I don't know if that answered your question.
Yeah, I'm just curious. Is it weather? Is it predators? Is it food?
That might be weather, because they are Palearctic species. They are usually found up north in higher latitudes.
Right, that makes sense. Thank you, Camila.
Thank you.
All right, I think that's thank you for your presentation, Camila. And now we can move to the last presentation from Jorge.
Okay, thank you so much for listening.
All right. Okay, last but not least of today's virtual session, I would like to introduce to Jorge Rua. He's one of our co-directors at REPU. He's currently a PhD candidate at Vanderbilt University, and he was a mentee of the biology branch of REPU.
Hi, everyone. Thanks for the invitation. Okay, I'm going to share my screen. How can I... Okay, let me know if everything seems fine. Good. Okay, great. Okay, hi, everyone. I'm Jorge Rua. As Kevin, I'm a co-director of REPU, but also I'm a PhD candidate at the Cortes Lab in Vanderbilt University, which is in the city of Nashville in the USA. So in the laboratory, we study the processes of DNA repair and DNA replication in human cells. And today I'm going to tell you about a protein that I've been studying for the last 5 years, which is called hemcis. And I will tell you about the importance to remove hemcis from the DNA to maintain genome integrity. So as I know, not everyone is aware about DNA stuff, so let's start with some fun facts. Do you know that each human cell has around 6 feet of DNA? And that is true. If you can take the DNA from one cell, put one end on the floor and stretch it up to your head, it will go up until 6 feet tall. We are all 99.9% alike, even if we come from different countries or ancestry, we're almost the same. And the human genome contains 3 billion base pairs of DNA. Now, when I talk about base pairs, it's going to be a word that I will use during this presentation. So here on I'm going to use the pointer here. Okay, so here on your right, you can see the normal structure of the DNA, this double strand helix, that it's bound to each other by these colorful bars that represent the base pairs. And they are called base pairs because they come in pairs. The molecule adenine always binds thymine, and the molecule guanine always bind cytosine. So they are always matching in this way. And you can also see in some articles just with letters, A binding T, G binding C. And the sequence of these bases are the ones that are going to code our genomic information and will tell our body how to work. And we have 3 billions of base pairs in our genome. Why is this important? Well, this actually makes things a little bit complicated because every time one of our cells divides, first, it has to divide all the genome, all those billion bases. And this is a scheme of how replication goes. So the double-strand DNA will be unwinded by this protein and helicase. So then we will have single-stranded DNA, and the top are in the bottom. That parental single-stranded DNA will be used by a protein called DNA polymerase, which will use it as a template to start to synthesize a new strand. How do they know which base introduce? Well, the DNA polymerase read this single-stranded DNA and said, if there's an adenine here, I will put a thymine. If there's a guanine, I will put a cytosine because of the matching. This process is very complex because you have to replicate billions of base pairs correctly every single cell cycle. Additionally, we have trillions of cell division happening during human lifetime. And to make things even more difficult, we have thousands of DNA deletions in each cell every single day. So how can we accomplish this? So there is a kind of animated video that a laboratory made using real protein structural information on how this process will look like. Let me change this. Okay, so in this video, this is an animation how replication works. Here you can see the double-stranded DNA that it's unwinded by the blue protein, which is the helicase. And here you can see single-stranded DNA, both. One single-stranded DNA will be rapidly replicated by this DNA polymerase that, as I mentioned, will match the bases and will generate a double-strand DNA. The other single-stranded DNA seems to be longer and stretches and twisted. It's because the polymerase, there's a different polymerase that replicates that other single-stranded DNA, and that it's called the lagging strand. All this scenario is what we call the replication fork, which is when a double-stranded DNA is separated, so the new single-stranded DNAs can synthesize and the entire genome can be replicated. So this is how it should look like. Okay. Oh, I cannot change it with this. Okay, so looks complicated, but as I mentioned, the DNA damage can make things even more difficult. There are different types of DNA damage. Here there are some. Mismatch, when the base pairs are not matching correctly. Base lesions, when one of these bases it's not present or it's modified. And we have bulky lesions, when there is a protein attached to the DNA that can stop replication. And this is when you break one of the strands, single strand breaks, and when you break both of the strands. There are different types of DNA damage, which is a common thing that if there are too much of them, they can induce diseases. And which is the most common one? Cancer, neurological diseases, premature aging, and some of the diseases that arise when you have too much of these lesions. That is why it's so important to understand how the DNA repairs. There is a machinery that in our cells repair those lesions, but when that machinery is not working correctly, we have more of these lesions, and that can lead to these diseases. For my talk, I'm going to focus on a specific type of lesion that it's in this group, the base lesions. And this lesion is called apurinic apyrimidinic sites. It's also called abasic sites or AP sites. And what is that? Because as you can see here, an abasic site is the absence of a base. I told you already that if there is a guanine, here there should be a cytosine, but there's nothing. If here there's a timing, there should be an adenine, but there's nothing. So the basic sites are very common in our DNA. We usually have 12,000 of these lesions in each cell every single day. So why is so common? Well, they actually can happen spontaneously, but also as an intermediate step for repairing other type of lesions. For example, this uracil shouldn't be here. So the cell to repair this will remove it. We will have an evasive site. This guanine, it's modified. So the cell will remove it and that will create an evasive site. That's why it's so common. But the question is, okay, we have many of these evasive sites. How does it affect to the cell? Well, I already showed you a video of replication forks and how the double strand and it's unwinded for induce the synthesis of the new strands. If in one of those single strands we have an evasive site, that can stop the activity of polymerases. So replication will stop, and we don't want that. The cell, to overcome this problem, uses other type of polymerase, like this green one. This is called translation polymerase because if it detects an evasive site, it will introduce a mutation and continue. Okay, replication continues, but then we will have a pro-mutagenic strategy, and we don't want mutations in our genome. So there should be a better way to overcome evasive sites during DNA replication. And that's where hemcis comes to our story. Hemcis comes from 5-hydroxymethylcytosine, and it's a very long name that we don't need to remember. We are going to call it just hemcis. And hemcis is very particular. We know that it's present at replication forks. And in the characteristic of the protein, we can detect this basic surface that allows its interaction with the DNA. And also we can see here a catalytic pocket. This catalytic pocket contains residues that are important for the activity of the protein. Those residues, you can see it here in this diagram. This E, that is a glutamic acid, the H, which is a histidine, and the C, that is a cysteine. The cysteine specifically will allow the cross-link of the protein with the evasive site. So this protein will use this cysteine residue to be covalently bound to the evasive site if it's located in single-stranded DNA. That's a very strong binding. Why is that important? Well, when hemcis cross-link to the evasive site, it protects the evasive site from these polymerases that introduce mutations. And I told you we don't want mutations, so that's good. Additionally, when hemcis protects the evasive site, it interferes the activity of other endonucleases like APE1, which can cut the DNA if there is an evasive site. So by hemcis protecting the evasive site, we prevent the breaks. So that sounds very useful. Now, how can we detect this? In the laboratory, a postdoc called Karim Moni could detect those crosslinks by biochemical assays. The DNA protein crosslink, it's also called DPC, and that's how we are going to call it, hemcis DPC. Here you can see, for example, in this band is just a DNA with an evasive site. But if we include the presence of the protein, the wild type form of the protein will cross-link to the DNA and will generate this DPC. So the band shifts because now it's heavier. When we mutate that cysteine that it's important to the cross-link, then there's no cross-link anymore and we don't see the DPC. If we mutate the part of the protein that allows interaction with the DNA, also we prevent DPC formation. So this is a biochemical way to detect the presence of the DPC. But in the laboratory, we wanted to go further. So another PhD candidate, Petra Thompson, in collaboration with another lab, they could resolve the crystallic structure of the crosslink. So here you can see the actual evasive site in green. This is the part of the DNA when there is no base, and how it cross-links to the cysteine of the protein. So this structure is called thiazolidine linkage, and it's like the real photograph of what we see here. So there's no doubt that hemcis can cross-link to evasive sites in single-stranded DNA. Is that important? because we have to be very critical always with science. We know that the crosslink exists, but is it important for the cell? And it looks like it is. For example, these are viability curves. The black one is just normal cells that contains hemcis. When you introduce potassium bromate that can damage the DNA, and you incubate cells with that DNA damage agent, the viability will be reduced because it affects the genome. But if hemcis is not present, as you can see in these two examples of the gray lines, the viability is so much worse. Like if hemcis is not present, the activity of this drug, it's more deleterious. This is with another drug, MMS, that also affects the DNA. When hemcis is in the cell, okay, the viability is reduced. But when hemcis is not there, the viability is so much worse, suggesting that hemcis activity is also important to maintain genome integrity. Okay, so far, we know that during replication, we can have single-stranded DNA. If there is DNA damage, that can remove bases and generate the basic sites. If hemcis is not present, some other proteins can break here, and we don't want breaks because that is bad for our DNA. If another polymerase continue replication, it introduces mutations, and we don't want this. That's bad for our genome. So hemcis will cross-link to the basic sites to prevent those activities, just protecting the basic site. But what happens next? I already told you that another type of lesions are bulky lesions, which is when a protein covalently binds to the DNA. And that's not good because, as you can see here, that also interferes with replication. So how hemcis can be useful and at the same time be bad for the cell? Well, Johannes Walter has a research group in Harvard University, and he has studied how DPCs, not necessarily hemcis, but other proteins that cross-link to the DNA, are removed during DNA replication. And he proposed two models. Here on the left, he proposed that if there is a protein bound to the DNA during replication, a protein called aspartame will degrade it. In a very simple way, we'll chew it, and it will degrade the protein. Another possibility is that the DPC can be modified by these ubiquitin chains in green, so then another complex called the proteasome can detect it and degrade it. In both cases, he's suggesting that DPCs are degraded by proteolysis. So that's something that, and he published this in 2019. So I thought, okay, maybe this is what is happening with hemcis. Hemcis cross-linked to the basic site, and then aspartame comes and removes it, or the proteasome comes and removes it. And that's how replication continues. However, in my hands, in the laboratory, when I tested the proteasome, it had kind of a mild effect, not very important. And in my hands, at least, aspartame didn't have any effect. So is there another possibility? Is it possible that it can remove itself? And of course, I didn't think about this possibility, but someone else did. Catherine Pauling, which was a grad student from my year that worked in a different laboratory here at Vanderbilt, she was studying hemcis version in yeast. So hemcis is the name in humans, but in yeast, hemcis is called JET-K. And they noticed the same thing, JET-K cross-linked to the basic sites, but she noticed that that cross-link can be reversed by itself. And that was very new. So I wanted to test if that was actually happening in human cells as well. Maybe that's why the proteasome was not relevant, or the aspartame was not important, because it's not being degraded, yet it's being reversed. So that's the hypothesis that I've been recently testing. hemcis-DPC crosslink is reversible, and that is important to maintain genome integrity. So here, hemcis comes, crosslink to the basic site, and just by itself, it will remove. No degradation here. But according to Catherine Almidon, she says that this is mediated by one of the residues of the catalytic pocket, the E, which is called E127. So I needed to test this. How do I test or detect hemcis-DPC in cells? So in a very simple way, this is the way I set my experiment. This is not pizza. This is a plate with cells, and they are synchronized. I'm going to incubate these cells with a drug called CD437. This drug increases the amount of a basic site, which means that it will increase the amount of hemcis DPCs. Then I remove the drug, and then I recover the cells in normal medium. I incubate it for 4 hours, but during those 4 hours, I will take samples to see how hemcis DPCs are removed. And then I do a radar assay to detect it. So this is what you can see. Here you can see that when in the mock treatment, so when there is no CD437, the signal of hemcis bound to the DNA is very low. After the treatment with this drug, you can see a lot of hemcis DPC. But when you recover during these time points, you can see that the signal goes down over time, suggesting that hemcis DPC is being removed, which is what we are expecting. This is a control showing that we have the same amount of DNA in each sample, so it's not a loading problem. How do I quantify this? If I put this image in a figure, you can see that after the treatment, time 0, we have more of hemcis DPCs, which then goes down over time. Another way to quantify this is by percentage. So if time 0 is the highest amount of DPC that I can have, that would be 100%. So then each time points tell me the remaining percentage of hemcis DPCs. So once I have this curve, I can test the hypothesis. Is actually that residue important for the reversal? So here is the glutamic acid in the catalytic pocket, and I'm going to change this glutamic acid to glutamine. So I'm going to change the E to a Q. So I will create cells that express not only the some cells expressing the wild-type version of hemcis, but also the mutant version of hemcis. So this mutant version should not be removed, or the reversal should be prevented. And the first thing that I check, okay, without any stress, does it actually increase hemcis DPC levels or makes them more stable? And this is the first result that I got. I noticed that when hemcis has this mutation, there is more hemcis DPC in the cells without any stress, without any drug, suggesting that possibly it's retaining hemcis attached to the DNA. Then what am I expecting? I'm expecting these two scenarios. Wild-type cells, wild-type hemcis should act normal. So after cross-linking to the basic site, the glutamic acid should induce its removal. So then that's how I can see that hemcis is removed over time. But in this mutant, I'm expecting that the reversal is not happening. So I should expect the same amount of hemcis DPC over time. And I tested this using the radar assay. So the same experimental setting by checking the normal cells and the mutants. In the blue one, you can see the normal cells. Again, showing higher amounts of hemcis DPC that goes down over time. With the mutant, we can see lower initial amounts of hemcis DPC, but they're stable over time, at least during the first hour, and then start to go down. And I told you another way to see this is by percentage. If time 0 is you can have the highest percentage in each cell, then in blue you can see how rapidly hemcis DPC signal goes down over time. Meanwhile, the mutant, it's more stable during the first hour and then goes down. So this is telling me that mutant is actually preventing this removal somehow during the first hour. And I saw this in cells. Then I needed to see if in vitro, this protein will have the same activity. So this is an in vitro experiment where I have the protein hemcis in blue cross-linked to this DNA. Then I will introduce a longer oligo containing a deoxy site. If there's reversal happening, this protein should jump from here to here and generate a bigger DPC because it has a bigger length. And this is what I saw. Here is the initial 20 DPC, but in the presence of this longer oligo with a basic site, we start to see how the protein jumps to this longer oligo because of the reversal. And we know that here there's no other protein, just hemcis and the DNA. Now, this is the wild type hemcis. The mutant hemcis should prevent this reversal, right? And this is what I saw. With the mutant form of hemcis, you can see that there's no reversal happening. This is just a control, but you can see that there is no reversal. So when I quantify, we can see that the mutant is also preventing the reversal of the crosslink in vitro. Okay, so seems like the reversal is real in cells or in vitro. Again, the most important question, is that relevant for the cell? Okay, so I made some viability assays. So in here, if you look at the left here, the blue and the light blue are normal cells. U2 cells are cells, wild type cells, and here's our cells that are complemented with the wild type form of hemcis. So both of them act normally. The more drugs you have, the viability will be reduced. The red line is an empty vector, which is when the cell doesn't have any hemcis. And you can see that in the red one, when hemcis is not present, there's a deficiency in viability, clearly, during these doses. And the mutant, which is hemcis, but with this mutation, also behaves the same. And I could see that using CD437, which is the drug that forms the basic sites, but also with other DNA damage agents like potassium bromate. Like even if the protein is present, because of the mutation, it behaves like the protein is non-functional or is not even there. That is telling me that preventing the reversal is also affecting the general function of the cell. Then I wanted to know, finally, I wanted to know if having much more of this protein can affect the cell. So besides the cell lines that I made, I overexpressed the wild-type form of hemcis and I overexpressed the mutant. And without any stress, I check how fast they grow. And the cells here that have the overexpression of the mutant grow so much slower, suggesting that if you have too much of hemcis not reversing, that can affect cell growing, which means cell replication. It's not because there's too much protein, because the overexpression of the wild-type form grows normally. And here's another way to look at it. Here's empty vector grows normally, the overexpression of the wild-type as well, but if you have too much of hemcis that cannot reverse the crosslink, the growing is very affected. So this is what I saw. Reversal seems to be real in cells in vitro, and that's important for good viability. But if the reversal is not happening, then that can affect cells. So those are actually my conclusions, that the mutant of hemcis stabilizes hemcis DPC over time due to the lack of reversal, and we have seen in cells and in vitro. If there is a stress, like potassium bromate, MMS, that can make the cells even worse if they present this version of hemcis, which means that it's failing to maintain genome integrity. And the overexpression of this mutant, it's enough to affect cell growing, even without any stress. So that's it. That's the lab where I work. I want to mention that actually there's another Peruvian in our laboratory, Gabriela Gonzalez. She couldn't be in this picture, so she photoshopped herself here. So Gabby, it's a great supporter also of REPU. She knows about the program. And well, I will be glad to hear any question if you have some. Thanks.
Thank you so much, Jorge, for the presentation. Does anybody has any question? Jaime?
Yes. Can you hear me?
Yes.
Oh, nice presentation, Jorge. I'd like to do some questions. Firstly, I would like to know from where the Hensis,
Hensis,
is
yeah.
right. Yeah, Hensis come.
Hensis what?
From where do Hensis come?
From where? Oh, the name.
I mean, this protein it comes from any kind of species or any organism.
No, well, it's actually a very evolutionary conserve. You can find Hemsis in humans. We express Hemsis. You can find it in yeast. You can have it even in bacteria. So Hemsis is a very conserved protein. It goes all along different species, even from prokaryotes, suggesting it's a very basic role if it's important to maintain genome integrity.
Yeah, so you bought it from.
No, actually, I didn't buy it. Obviously, the cellular experiments, we use the Hemsis that it's already in cells. We didn't have to add anything. For the in vitro experiments, we use a purified version of the protein, but actually that was purified by another laboratory. Yes, that also works with Hemsis. So purification of proteins can take time, as some of you know. So it was easier to just ask for it, and that helped me to do the in vitro experiments.
Okay, I see. Well, there's another method that you could know that if this protein is removed, or it's the common method for any protein.
For proteins that are removed from the DNA?
Yeah, for knowing if this protein is irreversible or reversible. There's the only way, or could be another methodology.
Well, that's actually, that was the question of my entire PhD. Like, if there's just one way to remove Hemsis, there's more than one. And in biology, you can see that it's difficult to always have just one answer. So it is possible that there is more than one way to remove Hemsis. It looks like reversal might be a way, because we notice that by itself, it can remove from the DNA, even without the presence of the protosome of Spartan. That doesn't mean that it's the only one. Even if my data shows here, you can see that, okay, it's the protein without reversal, the protein is stable after 1 hour, but then you can see that in 2 hours, it goes down anyway, and in 4 hours keeps going down. So probably reversal can help for the first part, and maybe then something else comes along, because otherwise this will be a plateau, and it's not a plateau. Somehow the protein is removed anyways. But at least this data suggests that reversal might be important at the very beginning.
This is good or bad, or perhaps irreversible protein could be better?
I mean, it is what it is. And it is. I think it's good in a way that knowing that the protein can remove itself, that means saving energy, saving some other proteins like the protosome. The protosome, to degrade a protein, first needs to ubiquitinate the protein, which means the activity of other enzymes. So it's a little more complex event. As you know, we cannot stop replication too much time. We have to keep going with replication. So it might have in some sense that the protein covers the basic sites, but it also knows that it cannot be there too much long, so it can remove itself instead of waiting for another protein to degrade it. So maybe reversal is actually something good to maintain the timing normal with DNA replication.
Yeah, in this case, I think the same. What do you mean with DNA stress?
When I talk about the DNA stress, I mean the presence of molecules or radiation that can damage the DNA. So I told you at the beginning that there are different type of DNA damage, different type of lesions that you can have. Well, they can come from different type of stress. Like UV light can break the DNA. That's a type of stress. Potassium bromate can oxidize bases. That's another type of stress. That's what I meant. Like agents that can't induce replication damage, and with that can stop or delay replication.
Yeah, because you mentioned that when your protein binds to the DNA, to the stress DNA, yeah, it has another kind of behavior, right?
Well, when this mutant, because these mutants, the cells are alive, they replicate. But when there is a stress, the mutant, the cells that express this mutant, they behave like there's no even protein. Suggesting that the activity of crosslink is as important as the activity of removing the crosslink.
So your protein just works for the normal DNA. I mean, not very mutant, because when it's mutant, there's no bind.
No, the mutant, this mutant, E127, it can cross-link the DNA. Yes, because
Okay.
the cysteine is the one that allows the cross-link. This glutamic acid is the one that allows the removal. And since I mutated it, that's what I'm preventing. So the data suggests that the action of remove itself, it's as important as the action of cross-linking.
Okay, I see. Thank you so much.
No, thanks for the interest.
Does anybody else has any other question? Okay, I have questions myself, and maybe they're a little bit high level, but is there any, does it exist a disease due to the lack of hemcis or overcrowding of hemcis in humans?
No, not yet. Not that has been identified by any correlation assay. Usually when they try to address the activity of one gene for one disease, like they check the genome of patients with that disease and see if there is a correlation with any mutation, but so far there's no. However, there are other, yes, not direct with hemcis, but hemcis, the importance of hemcis is that can form a DPC and then remove. There are some diseases that can occur for having too much DPCs. Not necessarily hemcis, other protein that binds to the DNA. But if you have too much DPCs, that can be related to some diseases. Yeah, okay. And that's already known.
So it's actually when there is too much.
Yes, when there is too much.
Uh-huh. And what are the diseases? I mean, what are they related to?
One of them is like premature aging. I don't remember the name of the syndrome, but it's related to premature aging, which is a very common phenotype with when the DNA damage response is, I don't know, like complicated. But for example, I mentioned that aspartame, it's a protein that removes DPCs. And in my hands, it doesn't do anything for hemcis. But for example, when the aspartame is not working, then you have too many DPCs, and that's when this disease comes along. So it's more related to aspartame because it forms more DPCs. But for hemcis specifically, there's no point mutation, for example, that is correlated to a disease so far.
Okay. The second question I had is that is there, and this is again a high level question, like can the hemcis or aspartans, or for example, be found in like a supplement like version that people can ingest and, you know, increase something in their bodies to regulate them? Is there such a thing?
No, To be honest, chemistry is not a very well explored protein. It might sound confusing when people talk in a regular day of talking about getting proteins. It's like a milkshake or something.
Yeah.
But in terms of chemistry is actually in very small amounts compared to other proteins, but it's all present in the nucleus, suggesting that that's where it works. And as I mentioned, chemistry is important for the crosslink, but also the removing of the crosslink seems to be as important. So I would assume that maybe having too much of it, that will maybe somehow delay the removal. I don't know. In vitro, in my cells, having too much of the protein, the wild type form with no mutations, seems to be normal. The cells behave normal. But one thing is having cells in a plate, and another thing is having something in your body.
Right.
So yeah, I think it's very well regulated, the crosslink and the removal. So I think any change of that, like having too much to crosslink or having too slow of the removal, might affect its activity.
Okay. Thank you, Jorge.
Yeah, you're welcome.
Okay. So if seems like we have no more questions as of now, it's a good time to stop and wrap up this session, the virtual session. Thank you, Jorge.
No, thank you. I'm going to stop sharing.
All right. Thank you, everyone who has joined the YouTube session and the Zoom link, and we really appreciate your participation. And with Jorge's presentation, that defines the end of this today's virtual session. And I would like to remind you again that this upcoming Saturday we'll continue with the REPU seminar, where we will have virtual presentations as well as in-person presentations. And this will be broadcasted over YouTube Live as well. Thank you so much for your participation.